The workshop at Kristineberg in Bohuslän, Sweden is the 5th biofilm workshop in a series that is arranged every second year to gather researchers in ecotoxicology and ecology working on microbial biofilms. This year the focus was on different omic approaches in biofilm research. Omic has got to do with the words ending in omics, for example metagenomics, the genetic information in cells. And biofilms are microbial communities, groups of microorganisms that are very sensitive to environmental changes. Biofilms are therefore widely used to indicate the biological status of a place. Here Susanna Cristobal, professor from Linköping University, with a lecture on the topic of metaproteomics.
It's extremely interesting. The main focus of this workshop is to bring very modern methods, the omics techniques, now in application and use them to answer severe environmental problems. For example, the monitoring the Water Framework Directive, or to analyze the modes of action of chemicals. Can we use these modern methods, the omics techniques, and do they provide more information than traditional techniques? Yeah, I think that's the main points.
How do you find this workshop so far?
I think that is getting very interesting. So we have very interesting lectures from invited speakers, giving an overview of the cutting-edge omics approaches. So it's great participation from the participants. So poster presentations. I think that is going very well.
What did you just speak about in here?
In the omic world, we have different levels. We look to biochemicals. And we have, of course, the DNA, the proteins, the metabolites. And my special focus in my presentation were the metabolites, which are composing all the organisms. And from the pattern of these metabolites, we can get some information on the status of the organisms. And we conclude on the stress situation, for example.
How do you find this workshop so far?
It's very interesting and very broad in the topics. Yes, it has been quite useful for me. Yeah, it overlaps with a lot of my research.
What's your research about?
It's about bacteria, microbiomes in plants, in the flowers. So I'm studying how the bacteria can change the metabolites in the nectar.
Did you learn anything yourself so far?
Yes, a lot of things. So a lot of ideas to apply in the new research projects. So new tools, new approaches, new pipelines, a lot.